One of the areas that our lab works on is on developing in vivo methods for the characterization and design of regulatory RNAs in bacteria. Although we now know that non-coding RNAs perform critical functions in all domains of life, they're particularly important to bacterial pathogenesis. Indeed, an important aspect of being able to invade a host cell is survival of changes that take place in the host environment. These changes include changes in pH that lead to changes in acidity, changes in oxygen conditions, iron, and nutrients. Therefore, bacterial metabolism needs to be constantly reinvented on their pathogenesis. It turns out that various classes of non-coding RNAs are key to this process. As a definition, non-coding implies that unlike mRNAs that encode a protein product after being translated, these RNAs typically do not, although now we know that some can encode for short peptides. These specific types of non-coding RNAs are referred to as small RNAs in bacteria. At the molecular level, bacterial small RNAs can perform a number of functions by targeting cellular mRNAs. In this cartoon, bacterial small RNAs are represented by a blue squiggly lines here, and bacterial mRNAs are represented by a black squiggly line. As shown in panel A, they can downregulate mRNA expression by binding and blocking the ribosome binding site. Here they block the Shine-Dalgarno region. As shown in panel B, they can upregulate mRNA expression by increasing the stability of the transcript through direct binding or by binding to a site where they can compete with factors that can cause early termination, like the Rho factor. As shown in panel C, they can also downregulate expression by directly destabilizing the mRNA upon binding or by causing mRNA degradation. An important aspect of a bacterial small RNA is that it can simultaneously regulate multiple mRNAs. Importantly, at the molecular level, this regulatory mechanism occurs via antisense hybridization, where base pairing typically occurs within a small footprint of 6 to 10 nucleotide regions. A main challenge in fully understanding how sRNAs work in vivo is their complex behavior. They can have multiple functions by affecting multiple mRNAs and therefore multiple metabolic pathways, and they can also be highly dependent on environmental input and external conditions for their own expression. For these reasons, it is highly difficult to predict which mRNAs an sRNA will target and how these base pairing interactions will occur in vivo. Therefore, it has been difficult to determine targets for naturally occurring regulatory small RNAs in bacteria. Our lab has become very interested in character. The idea here is that in reality, RNA molecules are highly structured, and therefore there will be entire local regions that will not be available or accessible for physical interactions with other molecules, let's say targets. Our thought was that characterization of the local availability of all these sites, represented here by dashed lines, along the molecule would provide a good clue about activity. As we can imagine, the notion of accessible regions, here shown as stretches that can bind an mRNA here in black, it's key for regulatory RNAs to be able to recognize and physically contact their targets via antisense hybridization interactions. Importantly, these availabilities or accessibility within the molecule could be highly dependent to the cellular environment and to environmental conditions. For the last few years, we've been developing tools to map entire regions that are truly accessible within RNA regulators in vivo. We started by borrowing from the synthetic biology community an mRNA construct that changes from a closed to an open configuration to produce a fluorescent signal by the opening and closing of a hairpin loop. Specifically, this construct is made up of an RBS, a ribosome binding site shown here in blue, an anti-RBS complementary region shown here in orange, joined by a linker region shown here in black that encodes the sequence for GFP. To link the presence of this fluorescent signal to the presence of accessible or available regions within an RNA of interest, what we have done here is added a probe region shown here in red that it's another segment to this mRNA construct. What's special about this probe region, shown here in purple, is that it's complementary to a particular region of an RNA of interest, shown here in gray, so that if it detects that specific region accessible for interactions, the loop within the mRNA would get disturbed, and therefore it would go from its closed form to its open form, producing GFP, leading to a fluorescent signal that can be measured. In the opposite scenario, if this new probe, now shown in red, does not detect physically an accessible region within the RNA of interest, it won't be able to bind, the hairpin loop would remain closed, no GFP expression would be produced, leading to very little shift detectable in fluorescence. A nice feature of this approach is that we can use it as a high throughput way to profile RNA accessibility in vivo. What we can do is make a collection of these probe segments by building them into different plasmids, where each of these probe segments binds to a different region of an RNA of interest, shown here as dashed lines. And with fluorescence outputs, we could measure the accessibility of all the regions within an RNA in a single experiment. We can do this by building these probe libraries of plasmids where we can transform into a cell culture. Each cell presumably takes in one plasmid and reports on one sRNA region. So what we have built is a reporter that via very convenient plasmid-based system can measure RNA accessibility along a sequence of interest in vivo by assaying stretches of about 10 nucleotides that can form physical molecular interactions, and importantly can capture the complexity of the unknown molecular interactions of cellular effects that lead to more or less accessibility locally within an RNA. This method has been very useful to our lab for various applications in RNA characterization and design. First, we have been able to survey a variety of RNAs in vivo, including tRNAs and other regulatory RNAs of interest like the CSRB, to understand availability of the regions and being able to conclude potential sites for interacting with. Second, we have been able to profile entire collections of RNAs in the presence and absence of specific cellular factors, since this assays can be run directly in vivo. We have done that simultaneously for about 71 small RNAs, where we collectively studied the effect of the HFQ chaperone on the accessibility of these RNAs and found out that indeed the sRNAs are showed increased accessibility overall throughout their conformations in the presence of the HFQ chaperone. Third, something that we're really excited about is our ability to use experimental accessibility data for novel target discovery. Specifically, we have successfully implemented this method to filter bioinformatics predictions by only considering for experiments targets that bind regions that show extremes in accessibility with our assays. We have had some success with this approach and have been able to experimentally verify novel mRNA target. Lastly, profiling molecular accessibility has also allowed us to redesign RNA regulators with a wide range of function. Specifically, as shown in the top panel, we can now profile relative accessibility of all regions within an RNA of interest to infer which ones could be more active. Next, we can then recombine in different proportions more or less active regions by treating these like independent modules. As shown in the lower panel at the right, we have used this approach to rationally design and engineer CSRB, small RNA regulator mutants that vary up to 8-fold in activity relative to the wild type. In conclusion, we have designed a simple, convenient plasmid-based method that allows profiling of hundreds of RNAs simultaneously, corresponding to thousands of local regions within these RNA collections. An advantage is the method is based on antisense hybridization, which could also be a mechanism that is highly transferable to other bacteria.